So for the most part, meiosis moves forward without any deviations or any complications. This is pretty impressive when you consider the number of crossing over events and the very complicated movements of the 2 cycles of meiosis. However, it doesn't always go as planned when it comes to the separation of chromosomes. Normal separation during meiosis results in euploidy. Euploidy is just the correct number of chromosomes that are going to be associated with the species. Since we are human beings, we need 23 chromosomes to be present in our sperm and egg cells, or in our gametes should have 23 chromosomes apiece. However, abnormal separation of the chromosomes may result in aneuploidy. Aneuploidy is when we have a change in the number of chromosomes present in the gamete. Aneuploidy most often results from nondisjunction, or failure of the chromosomes to separate properly. This can occur in either meiosis 1 or meiosis 2. And this can result in a monosomy, which is having only one of a particular type of chromosome, or a trisomy, which is having 3 of a particular type of chromosome. And we're going to look at some example of those. So here is how nondisjunction may possibly occur in a pair of homologous chromosomes. Over here on the left, we have nondisjunction. So when we tried to separate those homologous chromosomes in meiosis 1, they simply failed to separate. We didn't properly check our seat belts. So remember when I talked about mitosis, where we have a checkpoint built in within mitosis, where we basically go down the line, kind of like ride operators on a roller coaster, and we check that everybody is buckled in. Well, we failed to buckle in properly. And so the spindle fibers didn't properly latch onto this particular chromosome within the homologous pair. And so both chromosomes, both of pairs of the homologous chromosome ended up on one side. So a normal separation would have looked like this during meiosis 1. This could, if this happened during meiosis 1, by meiosis 2, we would see gametes that have absolutely no chromosome, or they're missing the chromosome completely, or they have 2 copies of that chromosome. Because when we separated the sister chromosomes, we had already had 2 copies there. If nondisjunction occurs during meiosis 2, where we fail to separate those sister chromatids, one gamete is going to end up with both copies of those sister chromatids, whereas the other gamete will end up with no copies of that particular chromatid. If fertilization were to occur and we were to receive a copy of the chromosome from a sperm, this is what would happen in each case. In the event that we would have a nondisjunction over here, we would see 2 gametes that end up with an additional chromosome. This is what's known as a trisomy, where we actually have 3 copies of that particular chromosome. Now, what we want is 2 copies. We want a diploid organism where we have 1 paternal chromosome and 1 maternal chromosome. But what happened over here is we ended up with 1 paternal and 2 maternal chromosomes. Same with this one. We also have a trisomy. Now over here, because there was no maternal chromosome present, we actually are going to end up with a monosomy, meaning that we are actually only have 1 of a particular chromosome. We should have a paternal and a maternal chromosome. Instead, we only have a paternal chromosome. Same thing over here with our nondisjunction. Here we have 2 normal cells, but here we have a trisomy where we have 2 copies of the maternal chromosome and a single copy of the paternal chromosome, and then here we have just 1 copy of the paternal chromosome, leading to a monosomy. So like I said, a trisomy occurs when an individual has 3 of a particular type of chromosome. In humans, there are only 3 trisomies that are 3 autosomal trisomies that are viable beyond birth. Now, an autosomal trisomy is a trisomy of a chromosome that is not the sex chromosome. Autosomal chromosomes are just there for the rest of the body. Sex chromosomes, we differentiate. So you have in your body, you have 22 different autosomal chromosomes, and you have your 23rd chromosome is going to be your sex chromosome. So when I'm speaking here, this is about autosomal chromosomes. So many times those chromosomal anomalies do not lead to viable birth, but in humans there are 3 autosomal trisomies that are viable beyond birth, and those are going to be trisomy 13, 18, and 21, meaning that there are additional copies of your chromosome 13, additional copies of your chromosome 18, or additional copies of chromosome 21. Only one of those trisomies is going to be viable beyond early childhood. So both trisomy 13 and 18 do not survive early childhood. Trisomy 21 is also called Down syndrome. It can be recognized by the following characteristics: short stature, eyelid fold, flat face, stubby fingers, wide gap between the first and second toes. Those are some of the characteristics that are associated with Down syndrome. So here is what trisomy 21 looks like. It's when we have an additional copy of this 21st chromosome. They believe that most of the symptoms caused by trisomy 21 are caused by having an additional copy of the GART gene, G-A-R-T gene, which leads to very high levels of purines in the bloodstream, which causes all of those remaining symptoms. Now, just because most of the autosomal trisomies are not viable, there are quite a few changes in the sex chromosome number that do lead to viable offspring. These typically result from inheriting too many or too few X or Y chromosomes. This is going to be more easily tolerated than autosomal trisomies, because if you are female, you receive one X chromosome from your mother and one X chromosome from your father. One of those X chromosomes will be deactivated and you will be primarily controlled by your one X chromosome. If you're male, you're going to have an X chromosome and a Y chromosome. At an early stage in development, that X chromosome will be largely deactivated and you will be predominantly genetically controlled by the Y chromosome. So because you're predominantly controlled by the instructions of only one of those chromosomes, it's more viable or more tolerated than having additional chromosomes, autosomal chromosomes, because you're really only going to be influenced by the presence of one of your X or sex chromosomes to begin with. So this typically happens during nondisjunction, during oogenesis or spermatogenesis. In our example shown, we showed nondisjunction with oogenesis, but it can occur in spermatogenesis as well. Turner syndrome is an example of changes in the sex chromosome number. So Turner syndrome is caused by a female with a single X chromosome. Typically, we see that individuals with the Turner syndrome tend to be shorter, with broader chest and widely spaced nipples. They can be of normal intelligence and function pretty well with hormone therapy. Klinefelter syndrome is caused by having 2 X's and a Y. This is going to typically lead to a male. So Klinefelter syndrome is a double XY male. It's going to be a male with underdeveloped testes and prostate. There may also be some breast overdevelopment. Typically also leads to having very large arms, legs, and large hands. They do tend to have near normal intelligence unless we see multiples of that X chromosome, so XXXY or XXXXY. No matter how many X chromosomes are present, the presence of the Y chromosome is going to render the individual male. There are additional changes in chromosome number and structure. So we've talked about changes in the chromosome number. There are additional changes to the chromosome structure that can lead to genetic anomalies. So not all chromosomal abnormalities are caused by meiosis. Some mutations are in chromosome structure. Some mutations can cause abnormalities in chromosome structure, and a number of these result from environmental agents like radiation, organic chemicals, or viruses. Sometimes broken ends of chromosomes don't rejoin. Mutations can occur. Changes in chromosome structure include things like a deletion. A deletion is just when one or both ends of a chromosome breaks off. There could also be 2 simultaneous breaks that leads to the loss of an internal segment. Duplication is just when we have a presence of a chromosomal segment more than once on the same chromosome. Translocation. we'll go through each one of these. Deletion is when one or both of the ends of the chromosome break off. 2 simultaneous breaks could lead to the loss of an internal segment, but we're going to see deletions most frequently at the edges or the tips of a chromosome. Duplication is when we just duplicate a section. D and D, so out of the original chromosome was A, B, C, D, E, F, G. This chromosome goes A, B, C, D, E, D, E, F, G. So we just have a duplicated section of the organism, or of the chromosome. Inversion is also possible. It occurs as a result of 2 or more breaks in the chromosome. So basically what happens is we'll see a break right here and a break right here, and when we try to put that chromosome back together, we accidentally invert the chromosome piece, which, it could cause the genes to occur in a reverse order in an inverted segment. It's not such a big deal as long as all of those genes are still present, but as we will see, the genetic sequence of DNA can greatly influence the types and functionality of proteins that are present. So we will talk about this a little bit later when we start talking about gene transcription and translation. Translocation occurs when we actually move a section. A segment from one chromosome moves to a non-homologous chromosome. Typically follows the breakage of 2 non-homologous chromosomes and just an improper reassembly. So again, we broke both chromosomes and we tried to reattach them and failed to do so correctly. There are a couple of syndromes that are caused by deletions. Williams syndrome is a loss at the end of the chromosome 7, which contains a gene for the elastin. There are translocation syndromes. A Largile syndrome is a translocation between chromosome 22 and 20, which can lead to congenital heart defect. Chronic myelogenous leukemia, blood cancer, is caused by translocation between chromosomes 22 and 9. So here's a quick summary of what we covered this chapter. We covered an overview of meiosis. We talked about sexual reproduction and how it reduces the chromosomal number. We form diploid cells become 4 haploid. A single diploid cell can become 4 haploid gametes. Homologous chromosomes are going to code for the same genetic information. Then we talked about genetic variation. We talked about crossing over events during prophase 1, independent assortment during metaphase 1. We talked about how fertilization can also increase genetic variability. Then we talked about the phases of meiosis. We went through meiosis 1 and meiosis 2. Then we compared meiosis to mitosis. We went through the life cycle, and then finally changes in chromosome number and structure. That is it for this chapter. Please let me know if you have any questions, comments, or concerns, or want to bring anything up in our discussion groups.